of this that's been taking place so far has been electrical communication. And electrical because we have ions of charged nature. We have cations that are rushing into the axon. And the movement of sodium as it moves all the way through is going to create that action potential all the way through. So now what we look at in the synaptic communication is everything that takes place between the neuron and the next effector cell, whether it's a muscle gland or a neuron. So before we get to that, we do need to talk about how all the neurotransmitters that are stored, we get neurotransmitters that are stored at the axon terminals, we have to talk about how those neurotransmitters are released. And then we hone in our focus on what's happening on the inside, which we refer to as the synapse. And what exactly is a synapse? It's a junction between two neurons. That's basically it. So what we've talked about was the action potential propagation. We're good there. And we get to conduct that action potential down to the synaptic terminal. And what's happening specifically at the synaptic junction in between two adjacent neurons? And when we look at what we're referring to here, this is typically called the presynaptic neuron. And this is the postsynaptic neuron. And this right here being the synapse. And this is how we're going to be able to get one neuron to communicate with the next. We have a site at which we have communication. We're seeing the two types that we have here. We have electrical and we have chemical. And chemical being the most common type. And that chemical communication that takes place comes in the form of a neurotransmitter. A neurotransmitter that's exocytosed at the presynaptic neuron and diffuses it down its gradient over to the next. And so you'll see we have chemical, which we're going to be talking about. We do have electrical, which I'll talk about in a later slide. If we look at some of the connection types, you can see that axons typically will terminate at another dendrite of an adjacent neuron. We've got axon to axon, and then we've got some that are axon to the cell body. But really the heart of it is looking at axon to dendrite. That's going to be our most generic example that we're going to be using. The axon, which is going to be the transmitting end, to the dendrites, which would be the receiving end of the adjacent neuron. The neurotransmitters that are released from the axon terminal, for the most part, can be excitatory or inhibitory. It depends on what those neurotransmitters are. Again, I haven't talked about how we release the neurotransmitter. I'm just talking about the neurotransmitters themselves. So looking at the chemical, on the chemical makeup, and we'll talk about the different types of neurotransmitters, can either depolarize or hyperpolarize the postsynaptic neuron. And if you look at excitatory neurotransmitters typically will typically bind to potassium channels. And that should make sense. If you get an excitatory neurotransmitter that binds to a sodium channel, causes sodium to go in, that should cause the inside to depolarize. If you're to take a look at other neurotransmitters that are bound to potassium, or sometimes chloride. So that allows potassium to go out, making the inside much more negative, hence hyperpolarizing a neuron. As well as chloride. Chloride going into the cell, making it more negative. Again, hyperpolarizing it, bringing it below its resting membrane potential. So if you have a neuron that secretes excitatory neurotransmitters, what's taking place, the depolarization that's taking place in the postsynaptic neuron is what we refer to as a EPSP, excitatory postsynaptic potential. Excitatory because it depolarizes, postsynaptic because that's the second postsynaptic neuron after the synapse, and potential because those are the changes that are taking place in that neuron. And what that's doing, if it's bringing it closer, if it's exciting that neuron, what it's doing is it's depolarizing it, bringing it closer to negative 55 millivolts, being our threshold. So excitatory postsynaptic potentials typically make a neuron much more sensitive, more likely to fire. On the other hand, inhibitory is the exact opposite. With those types of channels, here you see you're hyperpolarizing it. Just like what we saw with the refractory period, we saw we're below resting membrane potential. So it's going to take an extra effort to bring us back to resting and bring us up to threshold in order to elicit an action potential in the postsynaptic neuron. When we look at all the neural activity that's taking place on that postsynaptic neuron, what we're doing is we're looking at the additive effect of all of those neurotransmitters that are being released. And that term that we look at is called summation. Same term that we saw earlier, but summation in relation to what's happening in the synapse. And that is the sum of all excitatory and inhibitory postsynaptic potentials. So let me take a look at another example. Here, I can take a look at a neuron. Here's another neuron. And I've got one bigger neuron. So I've got two terminals onto one neuron. I can have one that's excitatory, and I can have one that's inhibitory. If this one creates a 15 millivolt stimulus, and this one creates a negative 15 millivolts hyperpolarization, this should sort of cancel each other out and leave this resting membrane potential back at where it's normally at negative 70 millivolts. So that's a concept of summation. And we'll be able to break down what summation is. This is one type of summation. There's another type of summation that we'll talk about. So looking at these, here postsynaptic potentials can either depolarize or hyperpolarize, which is not new news. This talks about a stimulus that is excitatory, which results in an increase in, depolarization. Here we see an inhibitory postsynaptic potential where it's IPSP. You add those two together, they cancel each other out. So the two types of summation that we can look at, we can work either with space, space or time. And let me use an analogy that I know works better than the ones I've used today. Think of somebody that's sitting next to you, and you're sitting there and you're trying to pay attention, and somebody starts poking at you, okay? And you know you're good, you're sitting there, you're fine, you're not excited, somebody's poking you, right? They can either, taking a look at the summation that we have, either spatial or temporal. Spatial is where you have the summation of postsynaptic potentials from different locations on the postsynaptic potential, or postsynaptic cell at the same time. That means you have two people poking at you, up until the point that you reach threshold and you fire off and you do whatever you want to do, okay? Now temporal is the summation of postsynaptic potentials at the same location at different times. So that's someone poking you repeatedly, up until the point that those summate and you reach threshold and you do whatever you do, whether it's a backhand or forehand, whatever it is, okay? So looking at these, you can see on the graph at the bottom right, here you see an example of spatial summation. You have stimulus number 1 and you have stimulus number 2. This is the example that I was talking about earlier. Two presynaptic neurons firing onto a postsynaptic neuron. And if they're both excitatory, you can get one, wait, this is spatial? Yeah, this is spatial summation. From different locations at the same time. This is the stimulus that would result from one. This is the stimulus that would result from the second. But you get those two to fire at the same time, and it brings us up to threshold. So this could be a 7.5 millivolt stimulation. This could be another 7.5 stimulation. Add those two together, brings us up to negative 55, results in a firing of an action potential. Now looking at temporal, on the other hand, this is where we get that additive effect of that one presynaptic neuron firing multiple times, and since that presynaptic neuron is still adding up, this is summation. Summation up to the point we get to threshold, we fire again. So looking at these, when we're looking at, and I'm going to skip the electrical synapses, because these are synapses where we have gap junctions. So we get the direct movement of ions moving through. This is purely electrical. We don't really cover this until we get into our cardiac cells. Cardiac cells talk about the movement of ions in between cardiac muscle cells. Okay? And the advantage of this is we're not relying on any molecular diffusion across that synaptic cleft. It's purely ions moving through those gap junctions. Again, we'll talk about that later. So now that you know the principles of postsynaptic potentials, and again, the main purpose of adding those potentials would mean firing of that postsynaptic potential, making it to its next effector target. Let's take a look at how we get those neurotransmitters released to elicit either an IPSP or an EPSP. The whole principle, and I kind of go over this in multiple slides. Let me just kind of give you the gist of what's happening. Let me move to the next slide. In this portion of the axon terminal, we're going to be looking at our third voltage-gated channel. I know we talked about sodium, we talked about potassium. The third one in this case is going to be calcium. And this is the only place where we're going to be seeing calcium, voltage-gated calcium channels in the neuron. When that action potential makes its way down to the axon terminal, we're going to be depolarizing in that area that's going to result in the opening of our voltage-gated channels. And looking at that calcium is going to come in, and that calcium is going to have one specific role. And what it's going to do is it's going to start that process of the release of neurotransmitter. So here you're going to see that once that, and let me show you what's taking place, and this is really just kind of everything that's taking place. Let me show you what's happening at this point. Calcium comes in, and it's going to dock with these vesicles. These vesicles are filled with neurotransmitter. And for this, you have to think about what we talked about last week. We talked about neurotransmitters, and in most cases are going to be some form of protein, or amino acid, peptide, or a protein that's manufactured with our ribosomes, our endoplasmic reticulum, and packaged by the Golgi apparatus into these vesicles that are ready to be released across the synaptic cleft. Calcium comes in. Here you can see calcium rushing in. You've got these active vesicles that are filled, and here's our cell membrane. You can see that calcium comes in and binds to these docking stations that are these proteins right along the intracellular side of the cell membrane. So you'll see these vesicles that are filled with neurotransmitter come in, calcium binds and undocks these two proteins. And it literally will just break that point right there to cause exocytosis of those neurotransmitters. So that docking that takes place allows that to be released. Here you see dynamin and clathrin. You don't have to understand that process, but here we take that vesicle and we recycle it for that process again. So that's all it is. If you were to look at where calcium comes in, calcium works exactly in that mechanism, just to release those neurotransmitters in those vesicles. That's all it is. So these vesicles, if you were to take a look at these vesicles, they're already ready filled. We've got three types, and which you don't need to know, but three types of vesicles. They're already ready to be filled. You've got neurotransmitters that are being remanufactured and filling up these vesicles, and then we've got a reserve tank of neurotransmitters. So we've got plenty of neurotransmitter to be released as long as we get calcium that's been entering the axon terminal. That's going to release these neurotransmitters to make their way across the synaptic cleft to bind to the channels that we talked about earlier. So if you get a neurotransmitter that binds to a receptor, and each of these neurotransmitters need to bind to a receptor that's part of that channel. So as long as that neurotransmitter binds to, you get that channel that opens up, and that either allows sodium in, or for an inhibitory neurotransmitter allows potassium out, or allows chloride in. So with that, now that we've understand the concept of how we release the neurotransmitters, let's take a look at the chemical nature of the neurotransmitters themselves. We've got lots of neurotransmitters that we've studied over the years. You can see the same mechanism that works. The release of neurotransmitter, the same mechanism, the release. The biggest difference is what is going to happen on that target, whether we're going to depolarize or hyperpolarize. If you were to take a look at what the effector cell is, if the next target is a neuron, we're going to result in another action potential that should take place on that neuron to move down the line. Okay? If you see the next, the effector being a muscle, the muscle is essentially supposed to contract. So we need to release this neurotransmitter to make its way across, either to release an action potential or a contraction if we're another muscle. Some other of these neurotransmitters rely on a second messenger system. And what I'm talking about that specifically is that sometimes one of these channels, you get a neurotransmitter that binds, causes an activation. We don't actually get an ion to move through. What it's going to do, it's going to elicit a change that's taking place on the inside that results in what's called a second messenger. And we'll talk more about this in our endocrine lecture, but that neurotransmitter doesn't allow anything to take place. We get communication that takes place on the intracellular side. Here we would see something called cyclic AMP. This is an intracellular signal. It only happens on the inside of the cell. So that's what we're talking about. We'll go into more about that in our later lecture. So looking at our first neurotransmitter, the most common one that we're going to be able to see in a neuromuscular junction, that's a neuron targeting a muscle, is acetylcholine. Okay? Acetylcholine in neuromuscular junction is for the most part excitatory, or it can be inhibitory, depending on the location of the receptor. So anytime you have acetylcholine that's being released as a neurotransmitter, you have to have an acetylcholine receptor that picks up acetylcholine. When you see, taking a look at acetylcholine, you'll see that acetylcholine is just an amino acid. You'll see the structure on there. What's interesting about acetylcholine is they first studied it way back when. It was back in 1915 when they first studied neurotransmitter. And they worked with cardiac muscle cells. And what do they do when they see cardiac muscle cells? They have it supported with enough glucose, supported with enough oxygen. You have these cardiac cells that are still beating in a petri dish. So one of the first things they did in order to study neurotransmitters is they tried acetylcholine. And what did acetylcholine do? It caused those muscle cells to beat faster. So they knew it was excitatory. What they also looked at is they looked at some of the other receptors on here. And if you take a look at the receptors, there's ways to block those receptors that would result in the interruption of that chemical signal. So acetylcholine needs to be released by the presynaptic neuron, needs to diffuse across the cleft, and it needs to bind to a receptor. Now there are diseases, and if you remember when I used that term channelopathy was the pathologies of certain diseases, the etiology of a disease, the cause of the disease that causes that some dysfunction in that system. You'll see the receptors could be interrupted at some point that would not cause that signal to take place on the postsynaptic neuron. So in order to stop that signal, I can either inhibit the production of acetylcholine or I could inhibit the receptors. It's like if I were to destroy cell phone communication, I can either destroy the cell towers or I could somehow destroy your cell phones if that works. Okay? So looking at that, this gives rise to some of the autoimmune diseases that result. And the first one of these are called myasthenia gravis. And typically what you would see with these people that have myasthenia gravis is it's a progressive muscle weakness disease. And the first thing you'll see is the droopy eyelids. What's happening with people with myasthenia gravis is that they get this progressive disease that starts with muscle weakness, that starts with the eyes, and eventually works its way down to more of the vital organs, and eventually will start to cause a heavy decrease in the quality of life. And in myasthenia gravis, the immune system comes in and destroys the receptors, the reception of the signal of acetylcholine. So what typically happens with these receptors, here you can see normal receptors. You have acetylcholine that diffuses across and binds to those acetylcholine receptors. Here you have your autoimmune disease that comes in and you get these antibodies that bind to the same sites where you have acetylcholine binding to. So in essence, this works. Going back to an earlier lecture, the antibodies bind competitively at the same active sites for acetylcholine. And if it binds to, acetylcholine can't bind to. And if acetylcholine can't bind to those receptors because they're blocked by those antibodies, then the contraction doesn't occur. So what do? They have certain drugs called anticholinesterase. And if you take a look at the term, choline, acetylcholine. ACE, right? ACE being an enzyme. Anticholinesterase is an inhibitor of acetylcholine. I'm supposed to erase that term. Acetylcholine, I'm supposed to fix that. Inhibits acetylcholine no, don't erase that. Acetylcholinesterase. Acetylcholinesterase is an enzyme, bless you, that breaks down acetylcholine in the synaptic cleft. So when we release acetylcholine, it comes in, and you'll be able to see on the next slide, a couple slides over, this is how we remove the acetylcholine. Now, if we can inhibit the enzyme that breaks down acetylcholine, then we have more acetylcholine in the area that can bind to some of the receptors and still create that contraction. Okay. That'll make sense when we get to a couple slides down. The next one are amino acids. And the two main amino acids you'll see are glutamate and aspartate. Glutamine is something that you would see that's excitatory in the central nervous system. Taking a look at those neurons, most of the neurons in the brain are excitatory whenever glutamate is involved. If you were to take a look at GABA, GABA is gamma aminobutyric acid, and that is an inhibitory neurotransmitter. And if you were to sprinkle GABA onto the neurons, you would notice in the brain that would be inhibitory. So what that does is with the IPSP, that stops the action potential from taking place on the postsynaptic neuron. And again, looking at some of the other diseases that we've ever looked at, when we studied ADHD, attention deficit hyperactivity disorder, we saw that glutamate receptors were linked to hyperactivity. So it was the inability to inhibit our glutamate receptors that resulted in excessive activity, okay? Hence the hyperactivity. Looking at schizophrenia. Schizophrenia is linked to the expression of extra glutamate receptors. And if we know glutamate is excitatory, some of the neurons are supposed to be inhibitory. So if I express increased glutamate receptors, that makes our inhibitory interneurons much more inhibitory, resulting in imbalance in the postsynaptic potentials that are supposed to take place. So you see that a lot of these balances, a lot of these diseases are all related to the channels themselves. Other ones that we're looking at are modified amino acids. Take the amino acids, you modify them a little bit, and now you've got two main types. Catecholamines is a term that I'd like you guys to be familiar with. Catecholamines are typically things that speed the system up, things that you'll be able to see that are excitatory. Norepinephrine, epinephrine. Another term for epinephrine is adrenaline, okay? And dopamine. So these are catecholamines that we'll be referring to quite a bit in our later lectures that stimulate the sympathetic nervous system. If you were to take a look at the role of norepinephrine, you'll see that it's evolved in arousal, awakening, regulating mood. If you take a look at epinephrine, adrenaline, that increases the fight or flight response. Again, you guys remember Pulp Fiction? What happened they did with, I forgot what's her face. She had an overdose. Yeah. They injected her with adrenaline right straight to the heart. Fight or flight response caused the heart to beat harder, caused the sympathetic nervous system to pick right back up. Look at EpiPens, for instance. EpiPens that I know that have gone up in price. EpiPens are supposed to stimulate the fight or flight response, which is going to increase the ability for air to move into the lungs. It causes bronchodilation for people that have anaphylactic responses. Dopamine, if you were to take a look at dopamine, dopamine is tied into that pleasure response area. And again, one of those things that if you look at those areas, it also is involved in skeletal muscle tone. So if you're looking at emotional response and then skeletal muscle tone, remember dopamine is also released. It's a neurotransmitter, but we've got neurons all over the place. Whether they're involved specifically in areas such as the substantia nigra, parts of the brain that we'll talk about later on. Dopamine has a response there. Dopamine has a response in other areas of the brain, the pleasure center. When we look at serotonin, serotonin is derived from a modified tryptophan. Okay. hear that serotonin is related to sensory perception, temperature regulation, mood control, appetite. It's all enhancing these things. While on the other hand, serotonin, depending on where those neurons are, can also affect sex drive. So it's not uncommon that people that take any sort of serotonin or any drugs to increase their serotonin can also result in decreased sex drive. So these things are heavy. When you look at certain drugs that affect neurotransmitters, they have varied side effects depending on what other areas of the brain they affect. Other areas that we can talk about, ATP, nitric oxide, don't worry about that. We'll talk about that later on. But just to give you an idea that we just don't have just amino acids, there's many other ones. Looking at other peptides, neuropeptides. Now we're taking amino acids.